My name is Patricia, and I'm a postdoc here in this lab.
My name is Joyce Morales, and I am the lab manager for an oral biology lab at the University of Florida. As a researcher, I conduct research, I conduct experiments, and I present the data to the principal investigator, which is Dr. Jeannine Brady.
Can you tell us who you are and your role in the lab?
I am Jeannine Brady, and I am the fearless leader and PI of the Brady Lab. Well, I hope it's a place where we do really rigorous science and also respect that people have lives. And I hope it's a fun place to come to work too. I tell people if they don't like coming in the mornings, they're in the wrong field.
The environment in the Brady Lab, I would say that it's very chill. We have a very open communication. I enjoy a lot the fact that we are a very diverse lab. Everybody is from a different country.
I mean, Dr. Brady, ask anyone in the department, they all know her, and she's a person that always asks questions. So people would say, how is it to work with her? Like, is she tough? Because she's really tough when you're giving a seminar or something like that. So I guess, I mean, here it's like we have to show that we're good enough to work for her, and yeah. I think we have a nice environment. We all try to help each other, and there are a lot of students that comes here and work for us. Me, I have two students, Kaylee and Jarrett. They help me with cloning, and we're doing a lot of DNA agarose gels.
There are student assistants that are working with Dr. Riviere, which are Emily and Albert, and they are involved mainly in the structural characterization of the proteins, so a lot of structural biochemistry. Yeah.
What do you think is most important for a research lab to be successful and how they help?
So I think the most important thing is not to be competitive. I think for a lab to be successful, it's important to help each other and give support.
A commitment to hard work, respect for the taxpayer dollar. All of our funding comes from the taxpayer, so we want to be thankful for that and also not waste taxpayer money. So even though it might be fun to play with experiments and ideas, we do have a directive of what we have been funded to do.
The amyloid project, it contains research in amyloid formation and in biofilm development. So Streptococcus mutans adheres to the surface of the tooth by creating biofilm, which is like a layer of bacteria. And there are many components that go into the creation of a strong biofilm. So one of them is the creation of amyloid, which is simply an organized folding of certain proteins. These amyloids are more or less the same, the ones that you can find in an Alzheimer's patient's brain. So we characterize these amyloids, we characterize the proteins that create these amyloids, that produce these amyloids. And one of the main aims of this project is to probably be able to develop an antibody against strep mutans amyloids.
What would that do?
That would create an immune response. Like if we do that, that will create an immune response against the amyloid of the bacteria, which are also one of the factors aiding in the biofilm formation. So if you have an antibody against that, then you can destroy the amyloid. In a very general way, how antibodies work, you can target the amyloid and get rid of it, and then you will impair the formation of biofilm in strep mutants, and then will impair the ability of the bacteria to adhere to your tooth. Exactly.